All right. Good morning. First of all, I'd like to thank the OMICS group and the session chair for the invitation to talk about our latest research on developing a new approach to enhance oral bioavailability of drugs with poor permeability. I'd like to apologize in advance because I have a sore throat. I may cough intermittently during the talk, so excuse me for that. So as you probably know, there are many determinants to eventually determine the oral bioavailability of an oral drug. And these determinants can include the solubility of the drug itself in a certain dosage form, then the permeability across the gastrointestinal epithelium, and of course the first pass metabolism that can occur in both gut and the liver. And this equation, the F oral bioavailability, basically is a final product of FA, which is the fraction absorbed, then times whatever is left after the gut metabolism, and also whatever is left after the hepatic extraction. So today we're not going to focus on drugs that has extensive first pass metabolism. That's a whole another game to tackle. We're mainly focused on the fraction absorbed and developing ways to increase that this FA value. I'd like to thank Dr. Buperthit for setting up the stage, because this is an important FDA regulatory recognized classification scheme based upon the physical chemical property and also the permeability of the drug. So basically, what it says is the permeability as well as solubility of your drug dosage form is very important to demonstrate bioequivalence or bioavailability, bioequivalence. And essentially, this equation FA, again, that fraction absorbed is a function of total amount absorbed divided by the dose. And it also is a function of permeability and the gut wall, and also the concentration near the gut wall. So essentially, the permeability determines this function, and solubility has a big contribution to determine the drug concentration and or near the gut wall. And today, the approach we develop are mainly applicable for drugs with a high solubility, so it's not dissolution limited, but somehow the fraction absorbed is mainly limited by its low permeability. So in other words, the case 3 compounds is our main interest. Here are some examples of case 3 drugs that kind of cover a large variety of therapeutic areas. So there's enalaprilat, which is right here, salicylic acid, acyclovir, melagatran, and pentamidine, furamidine, and eflornithine. So these are small molecule drugs that, because of the presence of either amine or carboxylic acid, they tend to have good solubility, but due to either the charge presence on their molecule, they have very low permeability across the lipophilic biological membranes. And there are strategies, different strategies to try to improve permeability of these case 3 drugs. Prodrugs is one of the probably one of the most widely and successful approach. For example, there's these ester prodrugs developed for enalaprilat, which gives this enalapril. So basically, it's an ethyl ester of this original molecule. You also have this aniline attached to the hydroxy group to form an ester. Of course, we all recognize this aspirin is a prodrug also for the salicylic acid. So these ester prodrug improves their permeability significantly and enable them to have a much better oral bioavailability. There are also other prodrugs, for example, zymolegatran, which was developed by AstraZeneca almost about 10 years ago. And it's basically a double prodrug. There's a amidoxine prodrug to the amidine and ester prodrug to the carboxylic acid. And then there's a parfuramidine, which is a prodrug of furamidine. They put a methoxy on both ends of the molecule to make it more hydrophobic. These are prodrugs, however, they failed in the clinical trials due to the toxicity introduced by these prodrugs. So the take-home message is the prodrugs are a great method, but sometimes you got to be careful because you introduce a totally new molecule to the body. So sometimes there can be unintended consequences of those prodrugs. Another approach, instead of covalently modify your drug that you want to deliver to the patient, another kind of a general approach. Instead of modifying the drug, you can try to modulate the permeability of the gut barrier. So those are typically called absorption enhancers. So to kind of better understand that process, you may know this, but just give you a general overview of the drug absorption process that's going on in the gut barrier. So you have apical side, basically the gut lumen side, and the basolateral side, or the blood side. You have these enterocytes connected by the tight junctions, and you can have passive, for a lot of drugs, you can have passive diffusion through the enterocytes. You can have a carrier-mediated, these are typically drug transporter-mediated uptake process. You also have receptor-mediated transcytosis that can occur. Then this tight junction there's a process going on called paracellular permeation. This is typically very limited because of the very limited surface area, and also the organization of this tight junction basically exclude a lot of small molecules from passing through those barriers. But those are going to be the focus of my research, and we're trying to develop ways to modulate the permeability of these tight junctions. So there, over the last maybe 3-4 decades, there has been a number of absorption enhancers studied by various investigators. This is not an exhaustive list of all enhancers, but these have some common characteristics. They're non-specific. They have non-specific target. Typically, they have general effect in damaging the epithelium. So typically, their effects are irreversible. So at the same time, they increase when they increase the permeability of a drug. At the same time, they can cause damage to the epithelium or GI system. So they tend to have a limited therapeutic usefulness due to the similar effective and toxic doses. Then over the last decade or two, people become more interested in enhancers that have specific targets. And this is due to the better understanding of that tight junction I was talking about. So a number of research characterized tight junction protein complex as well as the adherens junction protein complex. These involves the proteins such as claudins or occludins, which are connected to the zonular occludins and then attached to the actin. And these proteins work together to maintain the tightness of the tight junction. And the adherens junction is kind of different from tight junction. These are consist of E-cadherin proteins that involved in cell adhesion. And this typically mediate the initial step of a cell-cell contact, and these are present in both endothelial and the epithelial membranes. There are also studies to identify or elucidate the signaling pathway that can regulate expression or function of these proteins. So I'm not going to go into those details, but we have much better understanding of these tight junction as well as the adherens junction. One of the better or most better studied and most has most promise is this protein called zonula occludens toxin, or ZOT protein, which is derived from cholera bacteria. It's a 47 kilodalton protein that can reversibly modulate the tight junction by this protein kinase C alpha activation dependent pathway, leading to the actin reorganization. And over the last decade, people have identified the active a fragment called delta G, which reduced the size of the enhancer. And eventually, about 6 years ago, they identified the 6-mer amino acid called AT1002 that, however, didn't show any effect in vitro, but in vivo increased AUC and Cmax of cephalosporin after intraduodenal administration. But a lot of problems with these enhancers, their stability. They're subject to enzymatic degradation, so you have to put a lot of protease inhibitors along with your enhancer to prevent degradation in the GI lumen space. What I'm going to focus on today is another relatively new enhancer called E-cadherin peptide. If you remember, these are the cadherin proteins present in the adherens junction that regulate the initial cell-cell adhesion process. So this is a kind of a picture or the structure of the E-cadherin protein present in the between the cell and cell space. And they have this so-called extracellular domain, EC1 to 5, this repeats region. And they can interact with each other by a calcium-dependent homophilic interaction. And this is just one of many potential interaction that this is called a trans homophilic interaction. There's cis homophilic interaction, but I just call it kind of like a kebab interaction because it looks like the grill you have. So anyway, the idea of these E-cadherin peptide or ECPs, they are derived from this extracellular domain, and the assumption is when you have this peptide present, they interrupt the kebab interaction of these E-cadherin proteins. One of the first generation ECPs is called HAV peptide. It's a 6-mer, so 6 amino acid. HAV is a conserved domain on the EC1 domain. And these peptides reversibly inhibit adhesion of KCO2 cells and show that it can increase the permeability of mannitol, donomycin, the model drugs, in the MDCK and KCO2 permeability models. And also eventually it was shown that these peptides also worked in terms of enhancing brain penetration of mannitol and donomycin using in situ rat brain perfusion. So these have some promises, but there are also problems with this one, mainly just like the ZOT or the delta G and the stability of these peptides. So our work, we designed a new peptide based upon HAV peptide, and it's called ECP-11. And the aim is to enhance its GI stability and also investigate its in vivo activity in terms of enhancing oral bioavailability of K3 drugs. So I'm going to go over our data on this new peptide, ECP-11. So in terms of stability, we first looked at it's sort of a chemical stability and as well as enzymatic stability in this very artificial system. in the simulated gastric fluid, basically with some salt and low pH, in the presence of pepsin, the gastric enzyme. So with or without pepsin, or in the water, it's very stable. With or without pepsin, this peptide is quite stable over 4-hour incubation time and 37 degrees, so almost 100% remaining. So it's not degraded chemically or digested by pepsin. And this is the ECP-11 scrambled. So this serves as our control peptide. It's a scrambled version of this peptide. So it's also quite stable. Then we looked at stability in the rat intestinal mucosal scraping, which give you, complex, the complex enzyme present in the intestinal system. We looked at the peptide stability over 4 hours again, using this intestinal mucosal scrapings. Again, it's quite stable over 4-hour incubation time. And to demonstrate, we still have active scrapings. We looked at the stability of an endogenous opioid neurotransmitter peptide called encapsulin, which is supposed to have very low stability. And indeed, in this mucosal scraping, it has a half-life about less than 1 minute. And this is another version of this peptide. So basically showing these mucosal scrapings have enzyme activity, yet our ECP-11 is still quite stable. So in those systems. And then we don't need to really look at the plasma stability because we're mainly concerned with the oral and the enhancer for oral delivery of drug. But nonetheless, we looked at plasma stability. In humans, it's quite stable, both ECP-11 and scrambled version. However, in the rat and mouse, due to probably species difference in the plasma protein present, and it's least stable in mouse plasma. You can see the degradation that occur quite quickly. So if we ever want to develop this peptide for systemic administration, it looks like we've got to be careful with animal models, but it may have a good promise for human application if we ever want to go into that direction. Now I want to show some data that we collected for 2 case 3 drugs, eflornithine and pentamidine. And the reason we started with these 2 compounds is I have an interest in developing new treatment for neglected tropical diseases. Human African trypanosomiasis, or HAT, is one of those neglected tropical disease. It's a parasitic infection endemic to sub-Saharan Africa. It's fatal if not treated. Typically has 2 clinical stages. First stage, where the parasites are limited in the blood, basically. And then second stage, the parasites actually invade the brain, so you require a drug to enter the brain to kill the parasites. Then pentamidine is used for the first stage, so for the blood stage. And eflornithine is used for the second stage, so you have to deliver the drug eventually into the brain. And these 2 drugs, eflornithine and sorry, this is pentamidine, this is eflornithine. Because they're case 3, they have very poor permeability, and currently they have to be given by IM injection or IV infusion. And if you look at eflornithine, it's given by IV infusion over 30 minutes, and you have to give it 4 times a day for 14 days to treat those patients with stage 2 CNS infection of the parasite, which makes this drug very, very difficult use in endemic region of Africa. So just kind of a comparison in the infrastructure and healthcare system over there. And we really, really like to develop a way to change the dosage formulation from these bottles to pills. So we first looked at the permeability across this in vitro model, MDCK monolayer. So you heard this from the first talk. These are typically permeability model you can run in vitro. Using this transwell system, you have a monolayer of cells, you look at the permeation or the flux of the drug from A to B, apical to the basolateral. So eventually we have permeability on the y-axis and different treatment on the x-axis. We have vehicle controlled and scramble peptide didn't show any difference. Then we have up to 10-fold increase in permeability when we treat it with the ECP-11. This is the eflornithine. We see similar results with the peptide treatment. Eflornithine has better intrinsic permeability compared to the pentamidine. So that's why the base permeability is several-fold higher than the pentamidine. And we looked at another model, the KECO2, which is also another classical permeability model, and we saw a similar effect with eflornithine. We didn't run this experiment with the pentamidine, though. Then we move on to more of a in vivo model, so to try to see if we have any in vivo effect by the peptide. So it's a simple pharmacokinetic study in rats with eflornithine. We have to we gave the peptide 2 doses, so 1 and 1 hour before the drug dose. Then we gave the peptide and drug together and time 0, and then monitor, and then monitor do the blood sampling over the next 24 hours. And with eflornithine, without the peptide vehicle, you have some reasonable, decent oral bioavailability. So you have plasma blood concentration of the drug over time. And then with the scrambled peptide, you don't have any effect, which are the triangles. With this peptide presence in the formulation, and you have a significant increase in the Cmax and also AUC of the drug. Basically, there's about 40% increase in terms of AUC. So that can translate into the basically 40% oral bioavailability increase of this drug. Because of the drug has to eventually enter the brain, so we also took the brain out of those rats and 2-hour post-drug dose and looked at eflornithine concentration, and as expected, the brain concentration of the drug also increases due to the peptide effect. So there's about also 40% increase in both brain concentration and the CSF concentration of the drug. So then the next experiment we run is with the pentamidine, and similar experimental design to look at the pharmacokinetics of pentamidine with the peptide treatment. So without the peptide, you have a relatively low exposure of the drug because of the low permeability, and the minimal inhibitory concentration of pentamidine for the HAT, that parasitic infection is about 100 nanomolar to 0.1 micromolar, so we're right above or below that range without the peptide. But with the peptide, we can increase that substantially, so we have much greater exposure, and that's by about 4 to 5-fold. So right now, and we're encouraged by all these pharmacokinetic data, and we're collaborating with another group to run the efficacy model for the HAT, so we can test the effect of our peptide and hopefully to develop oral formulation to treat the HAT. So to summarize, we believe this peptide is a new absorption enhancer to modulate permeability of an intestinal tight junction, and this kind of important thing is this peptide has necessary GI stability to improve the bioavailability without using large amount of protease inhibitor, which can be a barrier for drug development. Future research will look at try to elucidate the mechanism of action in terms of interaction between the peptide and E-cadherin proteins, and try to look at the application to other case 3 drugs in addition to the pentamidine and the eflornithine. So last but not least, I'd like to thank my postdoc Si-Hang for obtaining all these preliminary data, and Professor Taruna Siahan from University of Kansas for first developing the HAV peptides. Then our collaborator from Swiss Tropical and Public Health Institute to run the efficacy model for the parasitic infection, and CPDD is a consortium that aims to develop drugs for those neglected tropical diseases, and also University of Kansas General Research Fund for providing the seed fund to start up this research. And thank you for your attention.
How many people have PAT? How many people are afflicted with PAT?
Currently, the prevalence of HAT is much lower than about 10 years ago. I think the last report, about 2009, was about 10,000 diagnoses in Africa. Back in late 90s, there was a kind of a upswing of the occurrence due to the war and the political instability in Africa during the 80s and 90s. So that was about 40 to 50,000 diagnoses per year. So it's compared to malaria, and this is a much, has much less prevalence. But nonetheless, the disease is fatal if these patients are not treated, and malaria has got a lot of attention, and this HAT is drug kind of under the radar. That's why it's so-called neglected tropical diseases. Yeah.
So I have two questions. One is very general. So this peptide going to open the last barrier for tight junction. How about the first two barriers?
Good question. Exact molecular event that took place during this modulation, we don't know. So that's a kind of a simple answer. But what we believe, or our hypothesis, is the adherent junction is the first event that occurred to initiate cell-cell adhesion. So we believe it's blocking the formation. So it's not necessarily breaking it up. It prevents the formation. So because the gut epithelial regenerates, so we believe this is it inhibits that formation rather than disruption, and that's why we give it 2 doses. So we have about 1 hour, 1 hour before the drug dose. So because we believe it's not immediate effect, so it may take some time for the peptide to have the best potency in terms of modulating the gut epithelium. But exactly timing, we don't know. That's also kind of a research we're doing to see, how long does it take for the peptide to have the most effect? Is it 1 hour long enough, or we need, a couple more hours?
Actually, my second question related to your regimen. So you dose 1 hour before, and after that you dose together with the drug.
Mm-hmm.
So, but for drug development later on, so then this regimen may not be feasible for the dosage for development.
Well, it's going to be more difficult for sure, and exactly in terms of from regulatory perspective, how that is viewed, I'm not sure. That's kind of maybe there need to be more conversation about that. But back to the kind of science. So that's our initial experiment, because we believe there's need to be a time, but whether that time is also needed, we don't know. So if we just dose the drug and the peptide together, do we see an effect? At this point, we don't know. So if there already an effect, if you just give it together, maybe you just instead of once a day, you can give kind of 2 same dosage with the drug and the peptide together, maybe with 4-hour separation. Maybe that will work. So you don't necessarily have to give 2 different drugs. You can still give the drug and the first dose. There need to be a consideration of that timing, which require a lot of research to find out. But I agree that's going to be more difficult. I don't know if that's a.
As a generic
We have looked at, well, there's a upper limit. So typically, if you look at tight junction, that space, that space typically will allow a molecule with a molecular weight about 3,000 to 4,000 Dalton to enter. And so that may be the upper ceiling. If you want to deliver protein therapeutics, this may not be the way to go. But for small molecules, we've looked at these two, and donomycin, mannitol, which is a kind of just a control. So this works for those small molecules that with a molecular weight less than 1,000 Dalton. Yeah.
So you mentioned rate concentration goes increase, yeah?
Yes.
So it means a peptide affect not only oral absorption, also distribution, and it can be dangerous.
It can be what?
It can be dangerous.
Good. That's a really good question. So this is a question raised by NIH reviewer as well. So, so if your peptide getting into the systemic circulation, you can open up all sorts of tight junction and cause potential, toxicity. So I didn't show it here, but in those experiments, we also collected the blood, and we also developed the assay for the peptide in the blood. So we tried to see if we can detect peptide after the oral administration. And to our kind of relief, we didn't see any of the peptide got delivered into the systemic, at least below our limit of quantification of our analytical type, which is about 1 micromolar. As you can see, it needs about millimolar, or the upper hundreds of micromolar to have an effect in our in vitro system. So that's a very, very good, good question, and also concern we have. But at least right now, we didn't see any of peptide in the systemic circulation, and so whether or not that's going to be going to have an effect on the body, from our experiment with rat, we didn't see any overt toxicity of those rats. They live just like any other rats. So.
My question is along the same line. I know there's some reluctance to use tight junction modification for drug delivery, not just because it could potentially impact distribution of the drug itself, but also antigen presentation and potentially triggering an immune response and chronic inflammation. I know there are some clinical trials currently using tight junction modifiers, but I'm not aware of any approved drugs that utilize this technology. I was just wondering if there's precedence for that.
Again, you're hitting my soft spots. Yes, these are concerns. I don't think there are any approved ones at this point, and actually not I'm aware of. So there has been a lot of problems with tight junction enhancers, especially, I have that long list of modulators with non-specific target, and they tend to cause, they have effect, but they also, in the same dose, they tend to cause toxicity issue. The hope is with a better understanding of tight junction complex and signaling pathway, this new generation of modulators, when they have more specific mode of action, you may have a therapeutic window that will allow you to use this enhancer to increase the oral bioavailability. And ZhuoWang has been kind of a new one, but has its own problem in terms of stability. But we're hoping these E-cadherin peptides with better stability, can we apply, develop them into more, better way to modulate tight junction.
Now let's give applause to Mr. Wang.
Thank you.